This is a tobacco plant, and this is a genetically modified version of the same plant. And today we're going to replicate this project, only instead of tobacco, we're going to use this beautiful morning glory. But in our case, we're going to dive a few more steps deeper into the ocean of plant engineering, and we're going to make the whole plant express this red color, just like this. If the engineering is successful, we will be getting a plant which will naturally synthesize a red protein and will hopefully change the color. This is the part 1 of this project, and we are going to begin the work by sorting the seeds into a growth media, which will give us an uncontaminated working material. It is always a great idea to soak the seeds into a lukewarm distilled water for a day or two at least. The coating gets softer and much easier to work with. And in the meantime, I'm going to make a plant growth media. For 100 milliliters, we need 0.45 grams of MS cook, 0.3 grams of sucrose, a gram of agar-agar for solid media, and I'm also adding a 6-BAP, which is a cytokinin hormone and is used to stimulate the cell division. Right after I autoclave my growth media, I'm pouring it into a sterile jar and storing it for later use. Clean, sterilize, repeat. That's all it is usually. Here we would have a laminar flow hood, but we kinda do not. My friend and I kinda DIY'd it, you know how it goes, and fans broke. So I gotta fix it, but in the meantime, no more flow hood, I guess. There, I'm sterilizing the seeds into hydrogen peroxide for half an hour. It's quite essential because if the working material is contaminated, media gets contaminated as well, which is basically a failing point of experiment. After that 30 minutes has passed, here I am washing already sterile seeds into a sterile distilled water to remove any residue of hydrogen peroxide. After a good 5 minutes of washing, I transfer the seeds into a media, and just a tip is to do as much as you can when it comes to staying clean and firm, because that's the determining factor whether you will see fungi growing into your growth media or not. There is nothing more frustrating than working all day on your protocol, making the media, doing all the preparations, keeping the sterility, and in the end, there are weird suckers in your MS media which basically kill the plant. They are happy, they keep growing, they keep dividing, but your plant is good and very much dead. Yeah. This is the part 1 of this gene editing and tissue culture project. Stay tuned. Part 2 is coming, where I will directly engineer the plant with an Agrobacterium. Keep in touch and wait for the part 2.